Okay, so welcome to this next video in which we are discussing in endocytosis, specifically Clathrin-mediated endocytosis. And in this video what we're going to talk about is Clathrin. Okay, right. So far we've talked about endocytic motifs, we've talked about adapter proteins, and now what we're going to talk about is Clathrin. Okay, and Clathrin is the protein responsible for the budding off of a vesicle. Okay, so let's have a reminder of what we've seen so far with the endocytic vesicles and the adapter proteins. Okay, so we started off with the plasma membrane here. Okay, and we had our target protein within the plasma membrane here. So this is our target protein. Okay, and we want to endocytose this protein. Now we saw that any protein which needs to be endocytosed will have a certain intracellular domain. So let's say this is the cytoplasm on this side of our plasma membrane, and let's say this is the extracellular fluid, the ECF, on this side of the plasma membrane. Okay, so any target protein for endocytosis will have on its cytoplasmic side some sort of sequence of amino acids which is exposed to proteins within the cytoplasm that will tell the cell, will tell the proteins in the cytoplasm that this protein needs to be endocytosed. And this sort of a sequence of amino acids which tells proteins within the cytoplasm, look at me, I need to be endocytosed. This is what's known as an endocytic motif. Okay. Right, so any target protein to be endocytosed will have this combination of amino acids which will be clearly visible to proteins in the cytoplasm, known as an endocytic motif. And we've seen examples of endocytic motifs. So the most important and famous example is asparagine, the single letter amino acid code for which is N, followed by anything. So there's no amino acid which we use X for, so X just means any old amino acid can sit in either of these positions. And then finally you have a tyrosine in position 4, so the NXXY combination is a endocytic motif. So let me just label up these single letter codes. N is the single letter amino acid code for asparagine. Okay, so the primary amino acid of the primary amide of aspartic acid. Okay, and X means anything can go in that position, so this just means any old amino acid. And then Y stands for the amino acid tyrosine. Okay, so that is an example of a endocytic motif. Another example is tyrosine followed by anything, followed by anything, followed by Phi. Again, Phi is not a single letter amino acid code. Instead, this means any amino acid can sit here as long as it is bulky and hydrophobic. So it needs to have a bulky hydrophobic R group. So what examples of amino acids do we know which have bulky hydrophobic R groups? Well, examples would be the amino acid phenylalanine, which is denoted by F. So phenylalanine would be an example of a bulky hydrophobic R group. Okay. Another example would be the amino acid which has single letter code I, which stands for isoleucine. Okay, so isoleucine again is an example of a large R group which is hydrophobic, i.e. it has no polar functional groups at all. It's just a non-polar molecule, and therefore it interacts best with other non-polar molecules and therefore doesn't interact well with water, which is a very polar molecule. Okay, right, so these are endocytic motifs. We discussed then that what's going to happen is adapter protein complexes are going to come and bind to this endocytic motif. Okay, right, so firstly, we discussed how these adapter protein complexes are recruited to the plasma membrane. Okay, so let's say we have our plasma membrane here. Here's our target protein in again with the endocytic motif, let's say here in the cytoplasm. And then we have a very important protein sitting here in the plasma membrane. So this is the protein Synaptotagmin. So this is Synaptotagmin. And the Synaptotagmin that's important in synaptic vesicles is Synaptotagmin 1/2. Now you might be wondering, well, what on earth is Synaptotagmin doing in the plasma membrane? It's supposed to be in the synaptic vesicles. Well, let's have a think about this. If we have our axon terminal here, okay, and let's say we have our synaptic vesicle here with our Synaptotagmin protein sitting in here, then how are we going to get Synaptotagmin into the plasma membrane? Well, when we fuse the synaptic vesicle membrane with the plasma membrane, i.e. when we exocytose the neurotransmitter within this synaptic vesicle into the synaptic cleft, what's going to happen? Well, let's draw what's going to happen. Here's this synaptic vesicle now fused with the plasma membrane, and here is the Synaptotagmin here of the 1 or 2 type, so Synaptotagmin 1/2. Okay, so it's going to end up in the phospholipid bilayer of the cell membrane, basically. So that's how you get Synaptotagmin 1/2 in the plasma membrane. And these two C2 domains here, so this C2A domain, which is nearer to the transmembrane domain, and this C2B domain, which is further into the cytoplasm, these are both still on the cytoplasmic side. So here is the cytoplasm of the cell. Okay, and here is the extracellular fluid. Right, okay, and these two domains here, these are C2A and C2B. Right, so we've seen that the core domain of adapter protein complexes comes and binds to the C2B domain of the Synaptotagmin 1/2 protein, and this is how these adapter protein complexes get targeted to the cell membrane. So let's draw our adapter protein complex. Remember, adapter protein complexes consist of a bunch of proteins known as adaptin proteins. Also remember that there are more than one type of adapter protein complex, and the adapter protein complex that is important for endocytosis at the plasma membrane is the adapter protein complex 2, the AP2 complex. Okay, so I'll draw this AP2 complex out here. Okay, so it's made up of 4 adaptin proteins, and I'll write that in a moment. So it's made up of 4, not like that, cross that out. It's made up of 4 adaptin proteins. Okay, now, these 4 adaptin proteins are, you have the alpha adaptin protein, the beta adaptin protein, so these are these 2 halves here, and then sitting in the middle here, you have 2 adaptin proteins known as the mu, so we'll have this one here as the mu, so that one in the middle there is the mu, and then the sigma adaptin protein. So let's colour these in to make it look a little bit more interesting. Okay, so in red here is the alpha adaptin protein outlined in red. Okay, right, there we go. And, whoops, sorry about that. And then in blue, we'll have the beta adaptin protein. So here is the beta adaptin protein in blue, or outlined in blue anyway. Okay, so there's beta adaptin. And in purple, we'll have the mu adaptin protein, which sits in the middle there. Okay, and in turquoise, we'll have the sigma adaptin protein, which sits in the middle there. Okay, so this overall is the adaptin protein complex 2. Now, the adaptin protein complex 2 has 2 main regions. It has this core domain here, which consists of the sigma and mu adaptin proteins, as well as these sort of normal bits of the alpha and beta adaptin proteins. These sort of bits that stick out over here, these are known as the ears, or the appendages of the alpha and the beta subunits. So these are the ears, or more properly, the appendages. And basically, although I've drawn it upside down, you can see that it kind of looks like Mickey Mouse. And that's a comparison that often is used for adapter protein complexes. Right, so the adapter protein complex 2 is going to bind to C2b domain of the synaptotagmin protein here. Okay, and we'll draw this in green. And what we have found is that there's a lipid component of the phospholipid bilayer that enhances this binding. So in the phospholipid bilayer, what you often find is PIP2 molecules, which for the basis of this video I'll just draw like this. So there are PIP2 molecules in the phospholipid bilayer. They have a very similar structure to a normal phospholipid, and they take the place of a normal phospholipid in the phospholipid bilayer. So here's a PIP2 molecule, and as I say, they are a normal constituent of the phospholipid bilayer. Okay, and PIP2 seems to enhance this binding between the C2b domain of synaptotagmin 1/2 and the core domain of this adapter protein complex 2. Okay, right, now, this interaction is merely to target the adapter protein to the membrane. What is now going to happen is that the adapter protein complex is going to bind to the endocytic motif of the target protein here in turquoise. Okay, and then what's going to happen is the appendages, these bits here of the adapter protein complex, are going to bind to the Clathrin in order to actually endocytose this target protein. But we'll continue this discussion in the next video.